Hi, my name is Geeta J. Kumar, and my project is on circadian metabolism in macrophages. I'd like to begin by discussing a commonly known process called our circadian rhythms. The most common process associated with our circadian rhythms are our rest and wake cycles, also known as our sleep cycles. The circadian rhythm is controlled by the hypothalamus in a specific region in our brain called the suprachiasmatic nucleus. In addition to this, the sympathetic nervous system also plays a crucial role in the regulation of circadian rhythms. And although the body ensures that circadian rhythms are at a constant pattern, there are numerous factors that can influence and alter these rhythms. Many of the common circadian disruptors can include stress, pregnancy, dietary choices, and the most common, the lack of sufficient sleep. These are important to note because one of our body's most vital systems, the immune system, has thought to be dependent on circadian rhythms to perform its day-to-day function. And when these circadian rhythms are altered, it results in a weakened immune system directly correlated to cardiometabolic diseases. Now, what exactly causes these cardiometabolic diseases? This can be attributed to specialized white blood cells in our immune system called macrophages. Macrophage cells are a special subcategory of cells called phagocytes, and they are given this name for their ability to perform a crucial process called phagocytosis. This is when they engulf and digest foreign pathogens or anything that is potentially harmful to our body. In addition to this, macrophages also have the ability to perform inflammatory processes as well. In macrophages, there are two distinct forms of inflammatory responses, anti and pro-inflammation. It was found in numerous past studies that pro and anti-inflammation were related to different types of respiration. Pro-inflammatory refers to a macrophage causing inflammation. This often occurs when a person would obtain a cut or wound that causes the area to turn red and inflamed. And pro-inflammation is thought to be related to glycolytic respiration, or the metabolic process of glycolysis in cells. Anti-inflammatory is the opposite, and it is thought to be related to oxidative respiration. This occurs when macrophages have to stop the inflammation, and if macrophages fail to halt inflammation, the resulting factor is a rise in cardiometabolic diseases such as diabetes. And diabetes is thought to be directly correlated to chronic inflammation, which simply refers to an excess amount of inflammation that is ongoing within the body. This causes detrimental effects because this excess inflammatory response can cause excess pressure, resulting in serious heart diseases. The purpose of this study was to determine whether macrophage metabolism and inflammatory processes are regulated with circadian rhythms. This leads to my research question, which states, what are the circadian changes in metabolism in macrophage cell? I hypothesized that diurnal changes in macrophage metabolism regulates their ability to function in the inflammatory response. In order to do my experiment, I used a study conducted by Balsalobra et al., researchers at the University of Genevieve. The protocol that was used in the study was altered to look at metabolic changes using a machine called the XF, or extracellular flux, instead of looking at gene expression with the PCR machine. I used raw 264.7 macrophage cells, and these cells are genetically altered to be almost identical to cancerous cells. This was done so that the cells are able to thrive better outside the body and so that they multiply much rapidly as well. As per the protocol, my experiment was also based on an idea of serum shock. And this occurs when cells are given a large amount of nutrients, in this case FBS or fetal bovine serum, and it causes them to reset their circadian clocks. We do this so all the cells follow the same circadian rhythms in order to obtain accurate data. This method was found in the following paper, which is what the experiment was based on. This graph was taken from a study that measured gene expression in macrophages to test for circadian rhythms. Although I was measuring circadian rhythms, this pattern was expected to be the result that I wanted as well. The first graph here simply shows the oxygen consumption rate in cells, and this was a good indicator of how much oxygen was used, and also is a good indicator of oxidative respiration in the cells because 90% of our oxygen consumption is actually due to mitochondrial respiration. And here we see that the ECAR values, also known as extracellular flux, refers to the acidification rate, and this occurs when this refers to a process called glycolysis in the cells, and this acidification happens because in glycolysis there is a process where pyruvate turns into pyruvic acid, and as that occurs more prominently, the pH goes down, as we can see in the graph right here. All of this data was combined and used to determine an optimal concentration of cells that could be used in our experimentation. Based on all of these, a concentration of 100,000 cells was chosen because 100,000 kept the normal pH level of 7.4, and also because the standard deviation of 7.4 was fairly low. After this, the second part of my experiment was to establish the serum shock. The serum shock was used to reset the circadian rhythms, as I mentioned before. In order to do this, the cells were placed in flasks for 2 hours in a growth media containing no cell nutrients or FBS. And a solution after this, a solution of 50% FBS was used to shock the cells in order to reset their circadian rhythms. 50% was used based on the experiment conducted by Balsilobra and his colleagues. It was also found through experimentation that anything higher than 50% could potentially be harmful and kill the cells. Soon after the cells were shocked, they were placed in an XF machine for about 24 to 48 hours in order to determine OCAR and ECAR values. Initially, it was chosen that 72 hours would be used. However, it resulted in our data becoming far too skewed because the machine could not contain enough moisture to keep the cells alive. Not only this, but the media the cells were in also became highly acidic, causing cell death due to cell waste. This is the graph that I created, which shows what treated versus untreated cells should look like based on the OCAR-ECAR ratio. The ratio of OCAR-ECAR shows which process was more prominently used in the time period. Bigger numbers indicate higher OCAR values, and smaller numbers indicate lower ECAR values. The fluctuating patterns shown in the serum shock in blue show that there is a fluctuation in the different metabolic rates. In the span of certain hours, we expect to see OCAR levels being much higher than ECAR levels. As I ran the experiment, we can see towards the end of the graph here that there is a clear indication of circadian rhythms forming as we see the graph slightly arching, which created a close sine wave pattern. And as we ran the experiment one more time, we can see that there is no indication of a sine wave pattern and that the graph shows more close to an exponential curve. As can be seen, the hypothesis was not supported from the experiment. The data did not show that there was a circadian rhythm, showing that there was no to little connection between metabolic rates and inflammation in relation to the circadian rhythms in macrophage cells. I hope to continue conducting research on this topic in the future to hope that, and there could be a possibility that this hypothesis could potentially be true if the experiment was tested on peritoneal macrophages, which are cultured freshly from the body rather than raw 264.7 macrophages in order to obtain better results.